Familial hypercholesterolemia, or FH, is a condition that results from mutations in the LDLR, ApoB, and PCSK9 genes that have an autosomal dominant pattern of inheritance. In some rare cases, a person can inherit two mutated copies of either the LDLR, ApoB, or PCSK9 gene, which results in a more severe case of FH that will usually appear during childhood. Another case of FH can be from a mutation in the LDLR AP1 gene, which is characterized by an autosomal recessive pattern requiring a mutated copy from both parents. People diagnosed with FH can be characterized by high levels of cholesterol, which is a fatty substance made in the body and obtained from foods that come from animals. The human body needs cholesterol, but people with FH have difficulty in disposing of excess, causing it to be disposed into the blood. This excess of cholesterol in the blood puts people at FH at an elevated risk for heart disease, and FH can also cause a buildup of cholesterol clumps in the tendons, eyelids, and corneas. FH occurs in roughly 1 in 200 to 1 in 250 people, making it the most common inherited condition that contributes to cardiovascular diseases.
23andMe tests utilize a SNP chip to analyze DNA. A limitation of this type of genetic testing is that it can only detect SNPs, not insertions or deletions. 23andMe tests for one variant in the ApoB gene and 23 variants in the LDLR gene, which are linked to having very high LDL cholesterol levels. This is associated with an increased risk for heart disease. The LDLR gene provides instructions for making the low-density lipoprotein receptor proteins, which binds to the primary carriers of cholesterol in the blood, or low-density lipoproteins. Low-density lipoprotein receptors sit on the outer surface of many types of cells, where they pick up LDLs circulating in the bloodstream and transport them into the cell. They are abundant in the liver. Mutations in the LDLR gene can reduce the number of low-density lipoprotein receptors produced within cells or disrupt the receptor's ability to remove LDLs from the blood. As explained by Medline Plus, people with mutations in the LDLR gene have very high levels of blood cholesterol. As the excess cholesterol circulates through the bloodstream, it is deposited abnormally in tissues such as the skin, tendons, and coronary arteries. Familial hypercholesterolemia greatly increases the risk of developing heart disease. Most of those with FH inherit one altered copy of the LDLR gene from an affected parent, as well as a normal copy of the gene from the other parent, and these cases are associated with an increased risk of early heart disease. The presence of variations of this gene greatly increases the chance of developing FH. FH also results from mutations in the PCSK9 gene, which is not tested by 23andMe.
The test for high cholesterol familial hypercholesterolemia, as known scientifically, include more than 30 different SNPs for genetic variation. The test will have SNPs from all three genes, allowing testing variation from each one. Our test will focus on adding genetic variations for gene PCKS9, which is not tested in other tests. The gene contains a protein that is responsible for familial hypercholesterolemia. Genetic variation is called RS2894212. Variant information comes from SNP and is valid because it is peer-reviewed sources. LDLR genetic variation is one of the best predictors of genetic risk, but other two genes can also create hypercholesterolemia. Variation changes the genome, which is dominant allele. Proteins made throughout the body in many different cell types. Variation affects the body because increasing amounts of this protein from PCKSL9 creates a degradation protein that eliminates LDL, which is always extracellular in the bloodstream. This source comes from a peer-reviewed paper.
Our test, unlike the 23andMe test, will test for variations in the PCSK9 gene as well as the LDLR and the ApoB genes. More than 50 PCSK9 gene mutations have been found to cause familial hypercholesterolemia. Mutations in these genes that cause FH appear to enhance the activity of the PCSK9 protein, causing LDLRs to be broken down more quickly. This reduces the amount of receptors on the surface of liver cells, so there is an increase in LDLs, resulting in high blood cholesterol levels. Mutations in the PCSK9 gene is one of the main causes of FH. There are many reasons someone should not get a genetic test for FH. Firstly, there is a relatively high likelihood that you will receive a false positive. According to the Family Heart Organization, a negative test result just indicates that the genetic cause of the patient's illness were not discovered using the available genetic testing technology and current knowledge, not that the patient does not have FH. If FH is clinically diagnosed, between 30 to 40% of patients may have negative test results. These findings could be false negatives, or the individual could have a gene mutation that isn't yet known to be harmful or disease-causing. Their polygenic hypercholesterolemia may also be present. Additionally, there is reason to believe patients would be at psychological risk due to privacy and discriminatory issues. While most persons are protected from discrimination in the workplace and with regard to health insurance by the Genetic Information Non-Discrimination Act, other types of insurance, such as life, disability, or long-term care insurance, are not covered or protected. Informing people that they have inherited FH could allow them to prepare for the fact that they are at a higher risk for heart disease in the future. This could allow them time to adjust their diets for less cholesterol since their body has a hard time disposing of it, and help them reduce their risk of developing a heart disease. However, the results of the genetic test can greatly increase stress levels, potentially even unnecessarily, because just because someone has a genetic variation does not mean that they will have any issues in the future or that they will develop the disease. They could change many aspects of their life to prevent getting a disease that they most likely were never going to suffer from in the first place. Furthermore, they could spend their time and money working to avoid getting the disease when they didn't have much of a risk to begin with. Also, genetic testing is not cheap, so some could spend a lot of money on the test just to have normal results. Some companies store the results of the genetic tests, which could also be a privacy issue for some.